welcome to today's webinar. I'm here on behalf of Biochemical Society and Portland Press. So this is part of the Biochemistry Focus webinar series. So the series works like a range of different topics that can range from molecular bioscience all the way to practical sessions that can support career development. Each webinar will give you an opportunity to ask questions, and those questions will come to us via text. We also welcome some suggestions, and you could also suggest speakers for the following webinars. And if you want more information, please go to our website. There are plenty of information in there. So my name is Vanessa Marensi. I'm a lecturer in pharmacology at Bangor University. And today we welcome Robert Adamu Shey. He will bring a very novel and interesting topic. So Robert is from the University of Buea, Cameroon. He teaches and he carries out some research. He uses computational tools for the rational design and development of diagnosis and to development of vaccines aiming at infectious diseases. He holds a PhD in molecular biology of the University of Libre of Brussels in Belgium, where he also completed a 2-year postdoctoral fellowship. So in 2020, Robert was awarded the WHO TDR Clinical Research and Development Fellowship. He also underwent a 1-year training focused on research-related projects. So 1-year related project management at the European Vaccine Initiative in Germany. So currently his research is it's focusing in the development of some diagnostic tools and also in the vaccines that using a bioinformatic approach. And he will be talking about his informatic strategies. The title of his talk is Immunoinformatics and the Keys to Developing Diagnostics and Vaccines for Onchocerciasis. And yeah, so I think, Robert, I hand it to you now. Looking forward to your talk.
Thank you very much, Chairperson, for giving me the floor. Thank you to the Biochemical Society for giving me an opportunity to present this work on the topic application of immunoinformatics in the development of vaccine and diagnostic antigens for onchocerciasis. So as the Chairperson rightly said, I'm Dr. Shey Robert Adamu from the University of Buea in Cameroon. My presentation is going to run through a general overview of what onchocerciasis is. I'm going to talk about the research work we're doing at the Molecular and Cell Biology Lab at the University of Buea in the context of vaccine and diagnostic development, and I will end up with some future directions. Onchocerciasis is a neglected tropical disease caused by the parasitic nematode Onchocerca volvulus and transmitted by black flies of the genus Simulium. The fly breeds around fast-flowing streams, reason why the disease is also called river blindness. The parasite has a two-phase life cycle, part of which is in the black flies, which are the intermediate host, and the human beings as the definitive host, just to indicate that the adult female worm can live in the human for up to 15 years. The clinical manifestations, which stem mostly from the adult worms, are nodule formation on the skin, which sometimes can be on the head and different parts of the body. And in the nodules, you have the adult worms, which can, like I indicated, live in the humans for up to 15 years, during which they produce millions of microfilariae juvenile worms, which can migrate to different parts of the body, especially the skin, the eyes, and the lymphatics, where immunological reactions from interactions between the parasite antigens and the immune system leads to inflammatory reactions that result in the clinical manifestation. Apart from the nodules and blindness, you can also have various forms of cutaneous lesions, including different forms of dermatitis, atrophy, and hanging groin. Apart from the health impact of this disease, onchocerciasis is also associated with a lot of psychological and socioeconomic impact, including stigmatization and discrimination, particularly those who have skin diseases. There is poor school performance in children who are infected because sometimes they cannot concentrate in class, and there is a high rate of dropout because sometimes the children are forced to drop out of school to take care of their blind parents or siblings. People have also abandoned farmlands for fear of the infestation by the black fly vector. And because most of these societies are agrarian, there is loss of personal and household productivity. Concerning the global burden, onchocerciasis is reported to be endemic in about 34 countries globally, with an estimated 21 million people infected, the majority of which who suffer from skin disease, and some other people are also blind or visually impaired. It's estimated that about 250 million people live in regions where disease transmission is ongoing. Due to the enormous burden exerted by the disease, a lot of control programs have been put forth, particularly in Africa, which bears about 99% of the disease burden. The first of which was the onchocerciasis control program, which ran from 1974 to 2002. And the main goal of this program was to control the disease in selected countries in West Africa. The program succeeded in reducing the burden of disease and also reducing the number of people who got blind. And the main strategy was areas of riverine areas using helicopters, as you can see on the picture. Another program was launched in 1995, overlapping with the previous control program, and it was called the African Program for Onchocerciasis Control. Unlike the previous program, which focused on targeting the vector, the African Program for Onchocerciasis Control focused on targeting the parasite itself, and it did this through community-directed treatment with a drug that was donated called ivermectin or mectizan. The African Program for Onchocerciasis Control has also reported some success, particularly elimination in isolated foci in Senegal and Mali. And the successes of these programs led to the creation of a new program in 2016 called the Expanded Special Project for Elimination of Neglected Tropical Diseases. Unlike previous programs which focused on control, this program actually focused on elimination, and not just onchocerciasis, but other neglected tropical diseases. For onchocerciasis particularly, EXPAND targeted the elimination of the disease in 80% of endemic countries by 2025, which is the year we are in at the moment. In order to be able to eliminate any disease at all, there has to be a means to know who is infected. And concerning the diagnostic approaches for onchocerciasis, the gold standard is the bloodless skin snip test, which involves doing skin biopsy and examining the skin under the microscope. The test is reported to be very specific, even though some doubts has been raised on this. Lately, sensitivity is really low, and sensitivity is also dependent on the intensity of the parasite and the disease prevalence in the region of interest. The test is also very invasive and bears the risk of transmission of blood-borne diseases. To improve on the sensitivity of the skin snip test, nucleic acid-based tests have been developed. However, for this test, there is a need for specialized training, and it also suffers from field applicability issues, since most of the regions that are affected are rural regions which sometimes don't have electricity. There are also immunological diagnostic tests, and currently the test that is recommended by the WHO is the OV16 test, which exists in rapid diagnostic test formats as well as ELISA. Sensitivity is moderate, very high specificity. However, it's reported that this test exempts about 15 to 25% of people who are infected due to genetic restrictions. It's therefore been recommended that there is need for continuous investigation on new antigens that can help improve on the sensitivity of this test. Another important tool that is needed for elimination of diseases are tools that can target the parasite to treat those who are infected. And with regards to onchocerciasis, antivectorial measures had been in use since 1950s, but the current approach to control is using antiparasitic measures. And at the moment, ivermectin is the mainstay to control, and it acts at glutamate-gated chloride channels. It's very effective against the juvenile worms, but not against the adult worms. A big challenge with ivermectin is that it's estimated that about 1.5 billion treatments are needed, this through modeling studies, up to the year 2045. Ivermectin is also contraindicated in persons who are infected with lualua, another parasitic disease which is highly prevalent around Central Africa. A lot of research has been done on other drugs, and doxycycline, which is an antibiotic, has also undergone clinical trials. It shows great activity against an endosymbiont bacteria, Wolbachia, which lives in the Onchocerca volvulus parasite. It shows macrophyllaricidal activity, but given that it has to be taken for up to 4 to 6 weeks, it's not suitable for the community-directed treatment. And there are also fears about the possible emergence of resistance, given that it's an antibiotic. Lately, moxidectin, another drug, was approved by the FDA. It acts like ivermectin. It shows superior activity to ivermectin, particularly on its microphyllaricidal activity, but it's also contraindicated in persons who are less than 12, and it's not yet available for community-directed treatment. And so with this in mind, it can be said that the challenges that EXPEN needs to surmount in order to achieve its goal are numerous, among which are the lack of reliable diagnostic tools, the fact that ivermectin is only microphyllaricidal, the fact that the drug is also contraindicated in some populations, and also issues regarding geographical and chemotherapeutic coverage of ivermectin. With this in mind, it's been reiterated that onchocerciasis will not be eliminated in Africa with the current control tools. And so what EXPEN needs at the moment are, among others, better diagnostic tools, more effective chemotherapeutics, particularly drugs that will kill the adult worms, and also vaccines. I've highlighted some of the tools which I'm going to talk about in this talk. Immunoinformatics is a field which is at the interface of computer science and experimental immunology, and it represents the use of computational tools and resources for understanding immunological information. Since 2016, a couple of papers have been published on the genome of Onchocerca volvulus and its sibling parasite which affect cattle, Onchocerca ochengi. These publications provide tremendous amount of data that can be used for the development of control tools for onchocerciasis. With this, I'm going to move into the work that we've been doing at the Molecular and Cell Biology Lab at the University of Buea in the context of diagnostic tool development. Initially, in 2018, between 2019 and 2020, we published 2 papers characterizing different parasite antigens, OV58 GPCR and OV28 CRP, in the context of diagnostics. But one of the things we noticed is the fact that even though these tools showed some promise in the development of diagnostics, specificity was a big problem. And so lately, we have shifted to the development of multi-epitope antigens, which have several advantages, including the fact that they offer a higher sensitivity. This is because with multi-epitope antigens, what you can do is that you can pick out epitopes from different antigens and link them up together, and this increases the chances of having epitopes recognized by antibodies even when they are at low levels. Also, there is the possibility of overcoming strain variation with parasites that are highly variable by looking at conserved epitopes. In addition to improving sensitivity, multi-epitope antigens can also increase specificity and reduce the risk of cross-reactivity, which is a major drawback with immunodiagnostic tests. In 2017, a paper was published by a group in Belgium focusing on the immunodominant motifs in the Onchocerciasis volvulus parasite that were recognized by sera samples from persons who are infected with Onchocerciasis. Among the 12 peptides which were predicted, we selected 8 of them, which showed sensitivities above 80% and specificities above 90%, and we used them to develop a chimeric antigen which we termed OVMAIN1. OVMAIN1 was cloned and expressed in the context of a maltose binding fusion protein, and we assessed its antigenicity, and we realized that the chimeric antigen was higher. The antigenicity of the chimeric antigen was higher than its constituent peptide. We then went further to express the antigen, which we tested with sera samples from persons who are infected with Onchocerciasis, a healthy sera from a healthy population in Africa, as well as a European control population, and we realized that the antigen shows statistically significant difference, and it could be further characterized in the context of developing a tool for Onchocerciasis diagnosis. This work was published in 2019. We went further to characterize our design antigen in comparison with the WHO recommended test, and we realized that the profiles of both tests were quite similar in terms of its reactivity with persons who are infected compared to the controls. However, we went a bit deeper into the individual serum responses, and we realized that the reactivity profiles for individuals who reacted positively with both tests were quite different. And what we did was that we went further to assess the diagnostic parameters of both tests, and we realized that OV16 and OVMAIN1 showed similar sensitivities of about 70%. And when both antigens were combined together in an ELISA, the sensitivity dropped. But when we look at the number of persons who tested either positive either by OV16 test or by the OVMAIN1 test, there was an increase in the sensitivity up to about 88%. We are currently further characterizing the antigens, particularly in the context of cross-reactivity, which is a big issue when it comes to immunodiagnostic test development. In 2022, we teamed up with the Global Health Institute at the University of Antwerp in Belgium to do a proteomic analysis of the adult onchocercial worms together with the microfilarial stages, both the somatic antigens as well as the secretory products. Our profiles show that a total of about 1,400 proteins were gotten from the different stages, which we further characterize screening for the antigens that have a signal peptide, and we ended up with about 9 proteins which showed low conservation across other nematodes. And this is important because we wanted to design a test that is highly specific for our antigen of interest. The 9 proteins that were selected underwent epitope prediction, and we predicted the antigenicity of the predicted epitopes, and we selected 9 of the epitopes which we used to create a chimeric antigen which we termed OVMCBL2, whose physicochemical properties are indicated on the board. We went further to characterize this antigen, and we compared its reactivity with people who are infected with other related nematodes, including Wuchereria bancrofti, Manzanella pestans, Brugger malayi, and Ascaris lumbricoides. Our profile showed that the test was 100% specific, 100% sensitive, and it showed zero cross-reactivity with any of the serum samples from persons infected with related nematodes. We also thought it important to characterize the antigen in the context of monitoring community-directed treatment programs, and we realized that the antigen reacted differentially between those who are infected and those who have been taking ivermectin over a period of about 20 years. And at the moment, we are further characterizing the antigen, grouping the people who have been taking ivermectin under smaller groups to see whether we can have a better picture. One of the things we further did was that we also assess the OV-MCBL0, 2, and OV-16, and similar to what we did with OV-MEN1, and we realized that both tests had comparative sensitivities, and when we combined the tests, there was also a drop in sensitivity. But when we look at the number of persons who tested positive for either of the tests, there was an increase in the sensitivity to about 91%. And this is very important in the context of current monitoring efforts by the WHO. And so one of the things we realized is that even though antibody-based tests using serum are quite acceptable, sometimes when you go to the communities, people are still very reluctant to donate blood for you to develop the test. And recently, there have been a couple of reports that have suggested the use of urine antibodies in the development of diagnostics for other neglected tropical diseases, including leishmaniasis. And so what we did was that we further characterized OV-MCBL02 with urine samples from persons who are infected with onchocerciasis, samples from a healthy population in Africa and a European control population. And what we realized was that for total IgG and for IgG subclasses, we could attain sensitivities above 80% and specificities above 90% for total IgG and all of the subclasses except IgG4, for which we had very low diagnostic parameters. We are also further characterizing this antigen, particularly in the context of cross-reactivity with other related nematodes. And so with that in mind, these are the papers we have published in the context of onchocerciasis diagnosis using multi-epitope antigens. And I'm highlighting my colleagues, Dr. Shinpo Munchili Kabiru and Dr. Yengo Benis, who led the projects that led to this publications on the diagnostic tools we have developed. And so with the work that we did so far in the context of diagnostics, we received a 60,000 euro grant from the Belgian government for valorization of some of these tools. We also received a $30 grant from the African Network for Research African Network for Research in Neglected Tropical Diseases. And one of the students from our lab last year completed a one-year Commonwealth split site at the University of Glasgow in Scotland. And so with that, I will just shift to talk about the work we are doing in the context of vaccine development. And so why is a vaccine needed? I've talked about the limitations of ivermectin, including the fact that it doesn't kill the adult worms. There is risk of drug resistance. Distribution is logistically complex and difficult to sustain. And also there have been reported contraindications in different categories of patients. A vaccine will be a potential game changer as it provides the ability to provide long-lasting immunity. It can help in breaking the transmission cycle, as well as it offers a more cost-effective and sustainable path to elimination. It can also help in protecting those who are exempted from ivermectin, including children who are 5 or pregnant women who have been reported to be a reservoir for parasite transmission. With this, a vaccine is an essential tool to move the current goal of onchocerciasis from control to elimination. Efforts for vaccine development have hinged on different reports on the development of protective immunity, including the fact that about 1 to 5% of persons who live in endemic zones and are exposed to bites of the black fly show some form of natural protective immunity. There is also reports of concomitant immunity where those who are infected cannot be further infected. There is also zooprophylaxis, particularly reported in the north of Cameroon, where people who are exposed to bites of black fly and bitten and exposed to Onchocerca ochengi, which is the cattle parasite, are protected against the human parasite Onchocerca volvulus. And a number of studies have also reported protection using in vitro and in vivo studies in animal models. A couple of antigens have been characterized, and at the moment, OV103 and OVRAL2 have undergone extensive preclinical trials and are planned for phase 1 clinical trials. Protective immunity in onchocerciasis is reported to be multifaceted, and among the things that have been reported to be involved in protection is the involvement of toll-like receptor 4. The role of CD4 and CD8 T cells have also been reported, as well as cytophilic antibodies IgG1 and IgG3. When you look at the multifaceted nature of the immune response, one of the things that you can see is that a multi-epitope vaccine candidate can be a useful tool, as through a multi-epitope antigen, you can have the flexibility to incorporate the epitopes that would elicit all of these responses that are needed for protective immunity. And so concerning the advantages of multi-epitope antigens in vaccine development, the first is that there is enhanced safety and reduced risk, as in your design, you can exclude epitopes that are associated with any undesirable effects like immune suppression, autoimmunity, or epitopes that can be toxigenic. You also have the ability to precise and rationally design your antigen to include just the epitopes that you want, including maybe B cell epitopes, CTL epitopes, and helper T cell epitopes. There is also the possibility to use conserved regions for variable parasites in order to elicit broad protection. Thirdly, there is flexibility and versatility. Conceptually, it's possible through multi-epitope antigens to design a single vaccine candidate that can protect against different parasites. There is also the ability to incorporate in building adjuvants like TLR agonists, for example, since some of these pattern recognition receptor agonists have been reported to be involved in enhancing immune responses. And also there is the possibility to increase the coverage of the vaccine by incorporating epitopes to multiple human leukocyte antigens. And so with this in mind, this is the pipeline that we use in designing our multi-epitope antigen. And the focus, particularly for onchocerciasis, is to include linear B epitopes, helper T epitopes, as well as cytotoxic T lymphocyte epitopes. And so this is just a highlight of what we do concerning epitope prediction. We use different tools to predict different kinds of epitopes, which we are able to link together to form the multi-epitope antigens, which is the focus of a lot of the projects that we run. And so concerning what we've done so far, we initially designed two antigens, which we term OVGKR1 and OVGKR2. And OVGKR2 particularly was designed using epitopes from antigens that had already undergone extensive preclinical trials. And one of the things that we did for both antigens actually is the incorporation of a toll-like receptor 4 agonist at the N terminal of our antigen, since toll-like receptor 4 is reported to be involved in protection. For OVGKR1, it was designed using epitopes from antigens that have not undergone preclinical trials, but there were antigens that were predicted to be immunogenic using immunomic studies. So with what we have done concerning multi-epitope antigens for vaccine development, we've been able to design 3 antigens. I've not shown the last one here, which we also did a kind of comparison with the current vaccine candidates that are planned for clinical trials, OV103 and OVRAL2, in terms of antigenicity, instability, and allergenicity. And we realized that the 3 antigens that we designed showed antigenicities that were either equal to or a bit superior to the antigenicities of the vaccine candidates that are planned for clinical trials. For OVRAL2, which is planned for clinical trials, one of the things we realized is that it's predicted to be unstable. And when we always with the antigens do some computational characterization, and this is just a picture that depicts the interaction between OVGKR1 and toll-like receptor 4. And what we showed was that the antigen interacts with the toll-like receptor with based on the binding energies and their affinity that were predicted. And so for OVGKR1 and OVGKR2, we went further to express the antigens, and we tested the antigen with serum samples from those who are infected with onchocerciasis, those who are living in endemic regions but do not have the disease, samples from those who are samples from a healthy African population, as well as those who are co-infected with Walwa. We also use sera from a European population as control. And we realized that there was a differential reaction compared to the control sera. A similar pattern was obtained for OVGKR2, and also similarly when we combine the antigen into a cocktail. And so with what we have done for onchocerciasis vaccine development so far, we've been able to publish 3 articles, one of which received Editor's Choice Scientific Report in 2020. And in 2024, that's last year, I had an opportunity to spend 2 months at the University of Cambridge in the context of the Cambridge Africa Arborada Research Grant. And at the moment, I'm doing a 2-year postdoctoral fellowship at the University of Bonn in Germany. And so while multi-epitope antigens hold a great promise for development of different tools, there are also challenges and things that need to be considered. The first is that there is need for a knowledge of the pathogen's genome, proteome, as well as the specific protective responses that are required in the context of vaccine development. There is also the potential for reduced immunogenicity, as most often with the design, you have epitopes linking them together to form the strings, which sometimes may not fold together, and you end up having the epitope presentation not as optimal as required. However, the use of linkers can help to minimize this problem. There's also the issue of immunodominance, where a single epitope in the design may be immunogenic to the extent that it overshadows the response of the other epitopes, and so it reduces the breadth of the immune response, which is what you don't want with vaccine candidates. And so with this, the take-home messages that I have from this talk are the following: multi-epitope antigens, OVMN1 and OVMCBL2, which are our diagnostic candidates, leads to an increase in sensitivity when used with OV16 serum ELISA. The antigens also demonstrate the, desirable diagnostic characteristics in the context of the current onchocerciasis elimination goal. Our vaccine candidates OV-DKR1, DKR2, and OV-TM-MEV1 are predicted to have higher or similar antigenicity to current onchocerciasis vaccine candidates. OV-DKR1 and 2 also showed seroreactivity with samples from persons who are exposed but not from a controlled European population. And what we intend to do in the future are the following: to evaluate the cross-reactivity of OVMCBL02, which shows some promises with urine ELISA, with urine from persons who are infected with other parasites, particularly Lualua, which is a major parasite in the which has serious implications on current onchocerciasis elimination efforts. We also intend to investigate OV16 using urine ELISA, since it shows some promise with one of our antigens. We also intend to express the last vaccine candidate with design and investigate its immunogenicity with serum samples from human cohorts. And lastly, we think it will be important to investigate the immunogenicity of our different vaccine candidates in mice model to generate more valuable information that can be needed in the context of current vaccine development efforts. And with that, I want to appreciate my supervisors and mentors, including Professor Stephen Gogomo and Professor Eric Achidi at the University of Buea, Professor Jacob Sobgui and Professor Luc Banam at the Université Libre de Bruxelles, and Professor Vincent P.K. Chitanji at the University of Buea. And special thanks also goes to the members of the Molecular and Cell Biology Lab at the University of Buea, especially Dr. Shinto Munchili Kabiru and Dr. Yengo Benis, who generated the diagnostic data that was presented in this talk. I also extend our appreciation to the funders of the different projects, including the African Research Network for Neglected Tropical Diseases, the Central Africa Clinical Research Network, as well as the Belgian Society for Higher Education and Research. Thank you very much for your kind attention, and I'm open to answer any questions you may have.
Hi, Robert. Thank you very much. This is very interesting. And I think I should highlight that Robert also has received the 2025 award, Early Career Award, for the work that he has presented today. If you want to continue this conversation, we can go to you can go online and continue on a Biochemical Society, either on Next or on Skybloom. And we want to thank everyone that's attending this webinar. You can type your questions. I'll try to go over them and pass them to Robert. So, but first, I think I have one question. Okay. So I'm quite curious, when you design your peptides, do you take into account like any potential glycosylation, especially when you are modeling or doing some in silico studies? Do you think that could affect or change or in any ways how the antibodies would bind to your antigen?
Yes, so when we're doing our prediction studies, we generally do predict post-translational modifications, including glycosylations, phosphorylations, and all of that. But one of the things we notice is that a lot of the antigens that we have designed, for most of them, they actually don't have any glycosylation sites. And for some studies that have been done on onchocerciasis vaccine candidates, it was reported that for a couple of the antigens, the immunogenicity didn't really matter whether the antigen was expressed in bacteria or in yeast cells. So it seems for most of them, post-translational modification didn't affect immunogenicity or the protective ability of the design antigens.
Thank you. So there's a question for you. I'll read it out.
Okay.
So is the multi-epitope antigen to be used in antigen detection or antibody?
Yes, so they are used in antibody detection assays, not antigen detection assays.
Okay. And then it says that to remember the disadvantages of the antibody test and how we will exploit this test in the development of an antigen test. So
yes, so generally antigen detection tests are the ideal, but at the moment it's been difficult despite several efforts. And so there is a continuous reliance on antibody-based tests, even though they have their limitations, including the fact that they cannot distinguish between those who are currently infected and those who have had a past infection, since antibodies can continue to circulate. But at the moment, what the WHO is doing is that for monitoring, they are using antibodies in younger children to kind of see whether the disease is currently it's still circulating in an endemic population. So if children have antibodies, that's a sign that okay, transmission is ongoing.
Okay, thank you. So if anyone has any other question, you can type in that box. Meanwhile, I'm gonna ask one more question. Okay. So in the very beginning of your talk, you've shown some of the peptides you've designed, and then you've shown some proteomic studies.
Yes.
So I might have missed like the samples and the subjects you have used in each different study, but could you find in your proteomic studies any peptide that are in line with your initial design?
So the proteomics that we did in collaboration with the University of Antwerp in Belgium was just to be able to identify particular peptides that we can further use, to get peptide that we can use to predict epitopes that we can actually use to design our own antigens. And so what we did was that with the work that we did with the University of Antwerp, we were able to identify nine potential antigens for diagnostics, which we use for epitope prediction, and the epitopes were linked together to create a novel multi-epitope antigen for diagnostics, which we have tested, and it's showing some good promise.
Okay, that's great.
Yeah.
So first of all, we acknowledge that you've done a wonderful work, and the question is, how do you consider the folding of the structure of the antigens you're studying during the immunoinformatic analysis for vaccine development?
Yeah, so normally for vaccine development, what we do is that when we have the various epitopes linked together using various linkers, we have the string of this string of amino acid residues, which is our multi-epitope. And what we do is that we do the 3D structure prediction, and we also perform docking. Like I said, most of our antigens, we design them with a built-in adjuvant, and so we perform docking. Like for I think OVGKR1 and GKR2, we had a TLR4 agonist at the end terminals. So we do docking between the antigen and the TLR4 receptor. And so we do perform 3D structure prediction, and we do protein-protein interaction analysis.
Thanks, Robert. So next question. Could you comment on the changes in appetite over time for the use of tools like this in the management of tropical diseases?
Could I comment on the use of tools like this?
Yeah.
Yes, I can say that over the years, we have seen a tremendous rise in the design of multi-epitope antigens, particularly for vaccines for several neglected tropical diseases, but also a couple of them for diagnostics. But one of the things I can say is that a lot of the studies that have been published so far are just the immunoinformatics part. And so a lot of the studies actually lack the wet lab validation. And the truth is that immunoinformatics, while it offers a lot of advantages, it's always very important to, after you design the antigens, to go to the lab and do some wet lab testing. So I think it holds a great promise, but at the moment, a lot of the studies have just limited themselves to the antigen design, but not the wet lab characterization.
Thank you. So next question. Could you clarify why you use serum from a European population as a control?
Yes, with the sera from European populations are just used as additional controls. Since they are non-endemic and they are not exposed. So mostly our controls are people who are living in the endemic zones but are healthy, but we just use sera from European populations as a non-endemic control, just to be sure that the antigens we design are not reacting non-specifically.
Okay, so these are all the questions now.
Okay.
So thanks, Robert.
Yeah, thank you.
So I'll continue just with the Biochemical Society highlights. So the Biochemical Society 2027 award is now open for nomination. There is a wide range of recognition and opportunities. The deadline for initial nomination is the 3rd of November 2025. And then finally, do join the Biochemical Society because it's a great opportunity. There are like benefits, which can include discounts for registration fees for events and courses provided by the society, access to a wide range of grants and bursaries, personal online access to 2 of their journals, and other benefits that are listed in the website. And this is it. Thank you very much for today. Have a good afternoon.
Thank you.
Thanks, Robert. Thanks, everyone.